Hello and welcome to Life Sciences Knowledge Hub, your one-stop shop for industry insights and analysis. I'm David and we're joined now by Richard Williams from Crescendo Biologics. Richard, thank you very much indeed for joining us all the way from Cambridge here in the UK. Tell us a little bit about Crescendo.
Thank you, David. Yes, so Crescendo Biologics, we are based in the Baberham Research Campus just outside Cambridge. We were founded in 2009 and we're a clinical stage biopharmaceutical development company working for making treatments for immune oncology. So we were founded in 2009, as I said, and the early days were really to establish our sort of transgenic mouse platform. And this is the engine, the discovery engine for all of our molecules that we develop. So this is a triple knockout mouse that contains human VDJ genes, which allow us to basically immunize a mouse against a variety of targets, and then we get affinity matured, fully human entities from the mouse that we can actually combine together to make a therapeutic protein. We focus in immune oncology because we think our platform fits exceptionally well for that sort of modality. Traditionally, a lot of full-size IgG molecules have done okay for many different therapies, but in immune oncology and for solid tumor treatment, they are underperforming in some areas, and we're seeing more of an emergence for novel biologics and bispecific antibodies. We have had a very sort of active collaboration profile, so we've got some key collaborations with companies like Takeda, Zylab in China, and more recently Cancer Research UK in the UK. But we also have a very active, growing internal pipeline, and that is really my role at Crescendo, is to support the external collaborations, but also to drive through the internal programs into development and into the clinic. So we were more. Sorry.
No, I was just going to say it's been an interesting journey there from 2008, 2009, then when you were founded 12 or so years ago. How do you describe then your approach to cancer treatment? I know you've got some human body technology in there that you work with. Just to expand on that for us a bit more.
Sure. So what we call the human body is really the smallest binding entity of a full-length IgG antibody molecule. So the antibody molecule is normally around 150 kilodaltons, and the human bodies are only 12 kilodaltons, so less than 1/10 of the size of a full IgG antibody, but they still bind exquisitely and specifically to these targets that we raise against in our patented mouse. What we can do with the human bodies is prepare them as multi-domain molecules, so we can bind to several targets at the same time, and we can format these so we can bind to a cancer cell or a cancer target, but then also we can get dual engagements. We can bind to a T cell and bring that T cell in proximity to the tumor, so we can enact and start killing the tumor. The small size of our human bodies are great, and there's other technologies like the VHH technologies that you get from llama and other camelids. You get really good tissue penetration and retention in the biodistribution with these small molecules. And we've done studies and we've got publications recently that show that not only can we get our human bodies to access and actually enter the tumor very efficiently, but they actually stay in the tumor and where they need to work, and they have a very good safety profile. So we've got some really exciting data on the human bodies.
And what do you put your success down to? I mean, you mentioned partnerships a moment ago there, various partnerships in the UK and also over in China as well. Is that one of the ways that you feel as though you've been able to achieve the success you have so far?
Yes, I think it's that's probably key to the early success of Crescendo. Is really a validation of the pipeline that we were working with. The human bodies, what I should also say, are incredibly versatile as well. So, due to their small size, they can be used for sort of topical administration. They can be used to generate antibody drug conjugates. So we have collaborations to show that in CAR T therapy. But our internal pipeline is more directed at making sort of bispecific or multi-specific molecules in the immune oncology space. The collaborations have been really good at bringing revenue into the company and also validating the technology whilst we progress our internal pipeline.
So exciting times then, exciting partnerships, and a good strong foundation of a technology there. Casting your mind forward, we've spoken about how you've fed on your journey so far and how you've been able to build the platform that you now have. The kind of now, near and far view, if you like. Where do you hope to be in, say, 2 years, 5 years time in terms of product development and business development?
Yeah, that's a great question. Actually, I joined Crescendo 2.5 years ago to really set up the CMC team and the contract manufacturing network. We've got a really exciting few years ahead of us. So our first molecule is in the clinic now, and this is a bispecific molecule that binds to prostate-specific membrane antigen, or PSMA, and a T cell engager. So it activates a T cell in the proximity of the tumor microenvironment. That'll be starting clinical trials now. We've got a whole franchise of similar molecules using the CD137 T cell engager and enhancer. And we can pair these CD137 human bodies with any tumor associated antigen that we like, and we have many coming through in our pipeline now. But we've also got a really exciting time ahead with our collaborations. So we've got some great collaborations with Takeda, also with CRUK and with Zai as well. So a really exciting few years ahead for Crescendo when our molecules progress through the clinic and we become a mature biopharmaceutical development company.
Exciting pipeline, exciting partnerships, as you say there, with a variety of bodies. Listen, Richard, thank you very much indeed for joining us today, all the way from Cambridge, and I wish you every success in the future. Thank you.
Thank you very much.